So first I want to thank TimePie for the invitation and for organizing such a wonderful conference. Today I'm going to talk about our recent work on longevity. So aging, as we all know, is a super complicated process. So before we ask how humans age, we want to focus on aging of individual cells. And specifically, we want to ask two questions. How do single cells age, and can we reprogram aging to promote longevity? Many damage and stress factors have been identified to contribute to cellular aging, such as chromatin instability, loss of proteostasis, reactive oxygen species, mitochondrial defects, and many others. It has been generally believed that all these damage factors accumulate gradually during the aging process, leading to cellular decline and eventually cell death. However, in each individual cell, how these damage factors change dynamically to drive the aging process remains unclear. For example, one possible scenario is that cellular aging could be driven by multiple independent damage factors that accumulate at different rates, leading to different causes of aging in different cells. In another possible scenario, cellular aging could be driven by integration of all the damage factors, leading to a common aging and death pathway in all the cells. A careful single-cell analysis is required to determine which scenario actually underlies the aging process in single cells. To this end, we study the replicative aging embedding yeast, Saccharomyces cerevisiae. This is a genetically tractable model for the aging of mitotic cell types in mammals, such as our stem cells. And yeast replicative aging is measured as the number of mother cell divisions before its death. And through over 60 years of investigation, yeast aging study has identified many well-conserved genes that influence longevity among eukaryotes. However, a major limitation in the field is the traditional method for yeast aging study require manual separation of daughter cell from the mother cell after each cell division. So basically, traditionally, when we want to measure the yeast lifespan, what we have to do is we culture individual mother cell on a petri dish and look at them under a very low-resolution microdissection microscope. And whenever this mother cell generates a daughter cell, you will use a needle to remove the daughter cell and count this is daughter cell number 1. And then wait for 90 minutes while the other daughter cell is generated, and then you will remove this daughter cell again, and this is daughter cell number 2. And the reason you have to do that is if you leave the daughter cell there, the daughter cell will quickly become mother cell, and then it will become, quickly you will lose the track, which is mother cell, which is daughter cell, how many generations the mother cell can generate it. So you can see, and to identify a mutant that changes the longevity, you have to look at like 100 mother cells throughout 3 days, and removing daughter cells every 90 minutes. This is tremendously labor-intensive and also very low throughput. And importantly, it does not allow tracking of molecular and cellular changes during aging. To overcome this limitation, we developed a microfluidic device which traps individual mother cells at the bottom of finger-shaped chambers, allowing them to bud continuously while the daughter cells are removed automatically. So let's focus on, zoom in on a single chamber and look at the whole aging process of a mother cell. So this is a mother cell, it starts generating daughter cells upward, and then it switch budding direction, generating daughter cells downward, switch again, start generating these super elongated daughters, suggesting the mother cell is very sick, and eventually they will stop cell division and eventually die. So in this way, we can track the molecular and cellular changes during the aging process of a long single cells. So using this approach, we found that isogenic, genetically identical cells under the same standard growth condition showed 2 different types of phenotypic changes during aging. About half of the cell, when they become old, continuously produce these elongated daughters until cell dies, while the other half of the cell produce more round daughters until cell dies. We call them mode 1 and mode 2 aging. These mode 2 cells showed a more dramatic slowdown of cell cycle rate and a shorter lifespan than mode 1 aging cells. We further tracked different organelles during aging, and we observed that the nucleoli became dramatically enlarged and fragmented in all mode 1 aging cells, but not in mode 2 aging cells, while the mitochondrial become aggregated in all mode 2 aging cells, but not in mode 1 aging cells. So these results really showed that different modes of aging specifically target different organelles. Mode 1 aging leads to nuclear olar decline, while mode 2 aging leads to mitochondrial decline. So for age-dependent nuclear olar enlargement is related to the instability of ribosomal DNA, rDNA, located in the nucleolus. And a major mechanism to maintain the stability of rDNA is chromatin silencing, mediated by a conserved lysine deacetylase Sir2, which repress the recombination and instability of rDNA. To monitor RDNA silencing in single cells, we introduced a reporter fluorescent reporter gene under a constitutive promoter to the RDNA region. Because the expression of the reporter is repressed by silencing, so a decrease of fluorescence indicates enhanced Sir2 activity and the silencing, while an increase of fluorescence indicates a reduced Sir2 activity and the loss of silencing. For mode 2 aging with mitochondrial decline, they considered that in human aging, the deficiency of heme, a major functional form of iron, may be a key factor for mitochondrial decline. And in yeast, the HAP transcriptional complex is required for heme biogenesis and mitochondrial function. To monitor heme level during aging, we used a nuclear-anchored infrared fluorescent reporter. The fluorescence of this reporter depends on a heme degradation product, biliverdin, and was found to correlate to cellular heme level. So a high IRP signal indicates a high heme level, while a low IRP signal indicates a low heme level. So now to determine how Sir2 and HAP pathways operate together to drive single-cell aging, we introduced both the rDNA-GFP reporter and the nuclear IRP reporter into the same cell to simultaneously track Sir2 and HAP activities during aging. Here in this slide, I want to show you representative movies of single aging cells and the aging trajectories quantified from these movies. Here in this 3D space, the Z-axis represents percentage of lifetime from 0% to 100%. The X-axis represents nuclear IRP signal, while the Y-axis represents rDNA-GFP signal. So now let's start with the mode 1 aging cell. As we can see, during aging, these cells show the gradually increased rDNA-GFP signal and the nuclear IRP signal. So now let's look at the mode 2 aging cells. Very different from mode 1 cells, which see a sharp depletion of IRP signal and a gradual decline of rDNA-GFP signal. Now let's look at the whole population of isogenic cells. Here, these red points are from mode 1 aging cells, and the blue points are from mode 2 aging cells. We can further project the initial states of all the cells onto a 2D plot. The 3D space shows all the aging trajectories, and the ending states of all the cells can be projected onto another 2D plot. From this result, we can see that genetically identical cells age. They began their lives with relatively uniform levels of two reporters. During aging, they divert quite earlier in life and progress towards two opposite directions. Mode 1 cells ended their life with a low Sir2, high HAP state. This is consistent with nuclear olar decline, while the mode 2 cells ended their life with a high Sir2, low HAP state. And this is consistent with mitochondrial decline. So these results really show that isogenic genetically identical cells age towards two discrete ending states with anti-correlative Sir2 and HAP activity, suggesting potential mutual inhibition between Sir2 and HAP that mediate this divergence in aging process. We further confirmed this mutual inhibition by genetic analysis, and in addition to that, both Sir2 and HAP also have positive autoregulation from the previous literature. Based on this circuit, we developed a simple mathematical model composed of two ordinary differential equations simulating how Sir2 and HAP change during the aging process, which is governed by mutual inhibition as well as positive autoregulation of Sir2 and HAP. To make things easier to understand, let's start with a system without mutual inhibition. So here, this is a Sir2 HAP faceplate. This is a vector field from model simulation showing how Sir2 and HAP change, how Sir2 and HAP level change over time. And these arrows point to where the system will move toward. And these blue horizontal lines are called the Sir2 narc lines. On this line, the rate of change of Sir2 is 0. The red vertical lines are HAP narc lines. On this line, the rate of change of HAP is 0. Where they intersect, the open circles are called unstable fixed points. These are the points the system will escape from. And the closed points are stable fixed points. These are the points that the system will convert toward. And once the system reaches this point, they will stop there. These are steady states. So based on this phase plan, you can see, depending on where the system starts, it will move toward 4 different steady states. The low Sir2 low HAP state, the low Sir2 high HAP state, the high Sir2 low HAP state, and high Sir2 high HAP state. So now in Y-type condition, the mutual inhibition, with the addition of the mutual inhibition, the straight narc lines are distorted. The mutual inhibition prevents the narc lines from intersecting at this high Sir2 high HAP region, and therefore eliminate one of the stable fixed points. To address the stochastic nature of aging in single cells, we then switched to a stochastic version of our model and computed a potential landscape by numerically solving the corresponding 2-dimensional Fokker plane equation. Each of the stable fixed points of the deterministic system correspond to a local well on the landscape, with the depth of the well reflecting its fate decision probability. The deeper the well, the higher the probability. So in this way, the aging process in each single cell can be viewed as divergent progression towards different wells on Sir2 and HAP landscape. The low Sir2 high HAP well corresponds to the ending state of mode 1 aging, while the low HAP wells corresponds to the ending state of mode 2 aging. We further examined the genetic perturbation of our circuit, and among which twofold overexpression of Sir2 showed very interesting results. In addition to mode 1 and mode 2 aging, Sir2 overexpression also generates a new mode of aging process that ended at a high Sir2 high HAP state. We call it mode 3 aging. These mode 3 cells turn out to be extremely long-lived, and it can retain a normal cell cycle rate throughout the entire lifespan, which is very different from mode 2 and mode 1 aging. So now we have, in our model, once the Sir2 level is increased by twofold, its increased activity can partially counteract with the inhibition from HAP, leading to the emergence of a fourth stable fixed point on the phase plane and a new well on the landscape. The progression towards this high HAP, high Sir2 stable fixed point corresponds to the mode 3 aging, long-lived mode 3 aging that we observed experimentally. So now we have a simple model that can explain and simulate the natural aging processes. So here comes our next question. Based on this model, can we reprogram aging to promote longevity? So today I'm going to focus on our most recent work, which is to rewire the Sir2 HAP circuit into a negative feedback loop and create a synthetic oscillator for longevity. So let's first go back to our model. We showed that in Y-type cells, the aging process of each individual cells can be viewed as divergent progressions towards different stable fixed point or steady states on the phase plane. So all these steady states are detrimental for the cell. The low Sir2 high HAP state lead to rDNA silencing loss and nuclear odor decline, while this low HAP state leads to heme depletion and mitochondrial decline. So the endogenous mutual inhibition circuit of Sir2 and HAP resembles a toggle switch that drives the fate decision and prolonged commitment to this detrimental steady state. So our idea is to rewire this mutual inhibition into a negative feedback loop, which could lead to sustained oscillations in Sir2 and HAP levels. Such a circuit could replace the this detrimental stable fixed point with a limit cycle on the phase plane, in which the system will keep moving along this black circle and will never commit to any of the detrimental steady state. And therefore, it can promote longevity. So, to guide our network engineering, we first developed a simple model of feedback circuit, which is composed of transcriptional induction of Sir2 by HAP and a transcription repression of HAP by Sir2. With appropriate parameter values, this model can generate sustained oscillations in Sir2 and HAP levels. We then perform Monte Carlo simulation to systematically explore the parameter space and analyze the dependence of oscillations on parameter values. And we found that oscillations were favored by a strong induction of Sir2 by HAP, a high capacity of HAP transcription, and a tight repression of HAP by Sir2. So this will serve as our design criteria for network engineering. So let's start with our endogenous system. To enable a strong induction of Sir2 by HAP, we replace the native Sir2 promoter with a HAP-inducible CYC1 promoter. In addition, we tag Sir2 with N-cherry so that we can track its protein dynamics. Then to ensure a high capacity of HAP transcription, we replace the native HAP4 promoter with a strong constitutive PDH3 promoter. Finally, to enable a tight repression of HAP by Sir2, we insert HAP into the rDNA region, which is subject to transcriptional silencing mediated by Sir2. So in this way, the Sir2 HAP circuit was reversed into a negative feedback loop. These are time traces of Sir2 abundance in representative cells. And we found that almost all of the engineers, the cells, showed oscillations during aging, while the Y-type control cells did not show such oscillation. And importantly, our synthetic oscillator can indeed lead to a substantially extended lifespan, with an 82% increase compared to that of Y-type control cells. This is the most pronounced lifespan extension that have ever been observed in yeast. So we compared our oscillator with the longest lived single or double mutants previously identified using genetic screens. And our oscillators has a longer lifespan than all of these long-lived mutants. And in addition, our oscillator showed a faster cell cycle rate than Y-type and other long-lived mutants throughout the entire lifespan, indicating that it has a healthier cellular state during aging. And based on our model, the oscillator can extend the lifespan because it can prevent the aging cells from committing to rDNA silencing loss or heme depletion. To test that, we monitored rDNA silencing and heme biogenesis in the oscillator stream using the rDNA-GFP reporter and the nuclear LP reporter that we developed previously. So here in this heat map, this is the Y-type condition. In this heat map, each row represents the time trace of a single cell. The color represents the fluorescent intensity, and the two plots shows the fluorescent changes of the two reporters from the same cells. And in Y-type cell, similar to what we showed before, about half of the cells are committed to mode 1 aging and ended their lives with continuous silencing of loss of rDNA silencing, while the other half of the cells are committed to mode 2 aging and ended their lives with prolonged heme depletion. In contrast, in our oscillator cells showed these short intermittent pulses of the two reporters throughout the entire lifespan, without a prolonged commitment to either rDNA silencing loss or heme depletion. So these results are consistent with our model demonstrating that our oscillator can enable a dynamic balance between rDNA silencing and heme biogenesis, and therefore it can promote longevity. So finally, to sum up, in this talk, we showed that genetically identical yeast cells under the same environmental condition can age through two different trajectories. One with rDNA silencing loss and nuclear odor decline, the other with heme depletion and mitochondrial decline. And we further identified a mutual inhibition circuit of Sir2 and HAP that serves as a toggle switch to drive this fate decision and commitment to these two aging paths. And we further then develop a simple mathematical model that can help us to understand the natural aging processes, and more importantly, it can guide the design of new interventional strategies. For example, based on the model, under the guidance of the modeling, we rewired the Sir2 HAP circuit into this negative feedback loop and engineered a synthetic oscillator for longevity. This work really demonstrates that we can rationally engineer the temporal patterns of aging processes by rewiring gene interactions, establishing for the first time the causal connection between gene network architecture and cellular longevity. And finally, I want to highlight the approach, the systems and the synthetic biology approaches that we used in this study, centered on a close couple of experiments and modeling, which hopefully will bring new advancements and innovation for aging research. And finally, acknowledgement, the first part of the work has been done by two very talented postdocs in lab, Yang Li and Yan Fei. And the second part, the synthetic oscillator, was done by another very talented postdoc, Zheng Zhou, and Zheng actually just started his lab in Shanghai, which is very exciting. So, and also the other lab members and our collaborators, Jack Ph.D., Lauren Pilas, Lev Simmering, and their trainees, and the funding sources. Thank you for your attention.
感谢郝楠教授的精彩分享，请您移步到舞台中央。下面我们开启提问环节，请导播老师上台切换设备。
Hey Nan, this is such a great work. I'm a big fan of your work. This is Dario Valenzano from the Leibniz Institute of Aging. So thanks so much. I have a question from the evolutionary biologist. So has this kind of like avoidance of fate commitments, this kind of like new circuitry that you engineer, ever been explored in actual cell biology, like in nature? Is this kind of like non-equilibrium state that you force your system to, is something that you can also observe in, I don't know, from data of like human cells, or are there physiological states or cellular states that resemble this type of thing naturally? Or is a completely new space of cellular circuitry that you guys are originally finding?
Yeah, this is a wonderful question. This is actually our current work, our ongoing work is to find, because the question is, this circuit is engineered, this oscillator circuit, and can we find some examples in the nature, or even in the yeast aging, that's driving yeast aging? So actually, the answer is yes, we are working on it, and actually we have some very nice preliminary data to show that actually there is some oscillation circuit in the yeast aging that are driving yeast aging. But the problem is this oscillator is not that robust. So that during aging, so let's consider this. If aging is a loss of homeostasis, the oscillation negative feedback is to maintain the homeostasis. So it is there at the first place. And during aging process, this oscillation fails, so that the cell age. So that's what we observe actually in our most recent work, which is not published yet, and we are still working on it, but we did see an oscillatory circuit there, which is slightly different from what we built, but the idea is the same. It keeps the homeostasis, and during the aging process, it failed. And we are thinking about using other approach to reinforce these oscillations and extend the lifespan. But that's a great question.
Great talk. Sort of along the same lines, I was wondering, in real situation, real aging cell, how many dimensions would you expect? Because here you have just two, right? But in reality, what do you predict? How many? Because we probably will have to balance multiple circuits.
That's another very, very, very good question. And again, another direction of study that is ongoing in lab. We are looking at the other, because now we are focusing on two pathways. One is silencing, chromatin stability, the other is mitochondrial decay. And we are looking at the other hallmarks of aging, the other pathways. protein homeostasis, and others. And some of the pathways are interestingly interconnected and can work with the two pathways that we found, and some of them are playing an additional role there. Definitely, there will be more dimensions. And however, but take a step back, although we say that there are multiple factors at the same time, but we reanalyzed the existing single cell RAC data, which is whole genome wide, to see the gene expression changes during the aging process. And what we see is consistent with what we found. We found two trajectories during the aging process, which is suggesting that they may be on the right road. So
that was a remarkably elegant body of work that you showed with the microfluidics and genetic engineering modeling, and it's really very impressive.
Thank you.
Now, one of the things that I'm not sure if everybody understands is that you're looking at a very rare mother cell, right? The oldest mother cell. And so, your data show that the number of replicative cycles is around 21, right? Mm-hmm. Which means you're looking at a phenotype of 1 out of 5 million cells, right? It's 1 over 2 to the 22nd power. And so this is not a selected trait, right? 50% of the cells in a culture are new daughters, have never been a mother, right? You knock out Sir2, there's no loss in fitness, right? And in fact, when Walter Longo deleted Sir2 and did the chronological lifespan assay, he showed that loss of Sir2, as well as calorie restriction, extend lifespan. So Sir2 is bad for chronological lifespan, really good for this replicative lifespan. Have you ever looked at the heme synthesis and the repression of ribosomal DNA circles in the chronological system? And do you understand why Sir2 is bad for cell survival and also good for the mother being able to replicate another time?
Yeah, that's a great question. So chronological aging, what we are focusing on now is called replicative aging. It's to counting how many daughter cells and mother cell can generate it. Chronological aging is another type of aging which we push the cell into a stationary phase and count how long they can live, the time they can live. So this is a very different type of aging process. And I'm not going to be surprised that Sir2 have a bad effect, because one of the central themes here is that this is always a double-edged sword, right? And the Sir2 has a good thing, but it repressed the heme biogenesis, which is bad. So we didn't look at chronological aging yet, but that's definitely what we can look at it. And the other thing I want to add to your question about all your comments about this rarity of mother cells. Because the idea is the newborn daughter cells, no matter how old the mother is, the newborn daughter cells are reciting their aging clock. So they are brand new. But based on some of our observational preliminary data, it is not true. Actually, the age state of the mother cell can affect their daughter cells. So it has a bigger effect to the whole colony. And I think Daria is also interested in this question. Yeah.
Oh, this is a fantastic talk, and we really need someone to have a deeper understanding of all aspects of aging, and this is wonderful. So I probably asked something that is probably so ahead of time, because this is single cell, of course, yeast. And so for the multicellular organisms, and we study cancer. So for most tissues, actually cells do not die or extensively, they live longer. It could be danger for cancer. So that obviously for multicellular organisms, I'm asking too much, but it is true that you have to much more dimension to consider. And sometimes the cell has to be limited in a sense. When they damage, they have to die. Otherwise, there'll be detrimental disease will happen, like cancer. Okay.
Exactly. That's a great comment. So the bottom line is this observation of this finding in yeast cannot be directly linked to human aging for sure. But scientifically, also we talk about cell senescence. which is also a kind of this aging process, replicative senescence. And again, it has good things, it has bad things. If you make the cell to replicate too much, it can lead to cancer. So senescence may be a good thing for preventing cancer. So that's very intriguing, the interaction between cancer and aging, definitely, yeah.
Yes, you could see that the evolutionary biologists in the crowd are very interested in this. Very elegant work. I wondered if the oscillator affects the replication rate rather than the total number of offspring, because nature's had billions of years to sort of perfect the yeast lifestyle. So curious if you've looked at that, or if you've ever tried to compete the wild type against the oscillator.
Yeah, that's another great question, and that's another ongoing work. Very good question. So the question is that why cells do this at the first place? Why cells have to diverge, right? Through evolution, it seems diverge is bad for longevity, right? And you can revere the circuit to make cells grow faster and live longer. Then by evolution, enough select that, right? I mean, that's a very interesting question. Our current hypothesis, based on some preliminary data, is that this divergence, this phase decision is age-dependent differentiation process that can lead the cell to specialized function. So it is, think about it, if we see the newborn cell as a stem cell, then during aging they will diverge, they will differentiate into special cell type with special function. And for our case, we see different stress resistance in these two different modes of aging cells. And a great experiment is to compete our oscillator cell, we can grow faster and grow longer with the Y-type cell. I think for the very nice condition, very rich condition, they may win. But in different stressful conditions which mimic the natural habit, they may lose. So that's the hypothesis. Yeah.
Really fantastic work. I'm interested in why they eventually die. So do they eventually, the oscillator just goes a bit too high and they commit to one of the lights, or is there another route to them to age that eventually kicks in that you don't normally see because they've only committed to those other things?
That's right. That's another great question. So, and another thing that we are studying, I don't think that they finally commit to the two types of cell deaths that we observed. The reason for that is that we can see for natural aging, we see cells slowly grow. The cell cycle becomes slower and the slower cell becomes thick, you can see it, and then they die. So you pretty much can predict when you look at their morphology, you look at their cell cycle. But this cell, this oscillator cell, which is very interesting, they can keep this young cell growth rate, cell cycle. You never can tell that they are dying. They are like young cell all the time, and all of a sudden they die. So there must be some other mechanisms that are causing the cell death, which we don't know yet. But that's a great question. There must be some other pathways that's kicking in.